Okay, let's dive into this. Welcome to our latest explainer. Today, we're pointing our microscopes at something that's literally happening inside you right this very second, millions of times over. We're breaking down the molecular mechanics of DNA replication. Now, it's super easy to just kind of think of this as, standard textbook biology, something you maybe memorize for a test and then just move on from. But I want you to imagine something entirely different. Don't just think of biology. I want you to imagine a fascinating, highly coordinated, microscopic factory operating at blinding speeds. We're going to explore exactly how this incredible molecular machinery works, using some fantastic source material to guide us through every single step of this biological marvel.
So, DNA replication, the biological process of creating an exact copy of a DNA molecule. Sounds simple enough on the surface, right? Just making a copy. But take a second to really appreciate the sheer scale of this. This is the ultimate biological copying mechanism. It's the absolute foundation that makes life on Earth possible. Every single time a cell divides, this exact, rigorous process has to happen with staggering fidelity. It's not just making a photocopy, it's preserving the very blueprint of who you are, molecule by meticulous molecule, across billions of base pairs without missing a single beat. It's just wild when you really think about it.
And this brilliantly illustrates the heavy lifting of our microscopic factory. Meet the absolute powerhouse of the whole operation, a crucial enzyme called helicase. Now, this is by no means a passive, gentle little event. Helicase actively consumes energy. Specifically, it uses ATP as its primary fuel source. With that massive burst of chemical energy, it powerfully drives its way forward, physically moving along the DNA molecule. Then comes the truly dramatic part. It forcefully unzips that famous tightly wound double helix structure, tearing the paired strands apart to form what scientists call the replication fork. It's literally like a high-speed locomotive plowing straight through a set of train tracks, completely dividing them in two as it just roars ahead.
So what actually happens to those tracks once they're violently split apart? Well, on one side, we start with that beautifully intertwined DNA double helix that we're all so familiar with. But once our powerhouse helicase does its job, we get something entirely new: separated single strands. And here is where our factory assembly line metaphor really comes into play. Because these separated strands, they don't just, float away into the cellular abyss. No way. They immediately take on a crucial new role. They act as precise templates. I really want to emphasize that word, template. They serve as the exact blueprint, the master mold, for building a brand new complementary strand of DNA. The raw materials are ready, and the factory is now primed for production.
But wait, let's take a beat here. Think about the physics of what we're discussing. What actually happens when DNA is forcefully unzipped at such staggering speeds? Think about a tightly tangled phone cord, or maybe a thick piece of twisted rope. If you grab it right in the middle and aggressively pull the strands apart, what happens to the rest of the rope ahead of your hands? It gets tighter. It coils up intensely into a state scientists call supercoiling. This right here introduces the great biological conflict in our factory narrative. Unzipping those strands creates intense, downright dangerous physical tension on the rest of the DNA molecule ahead of the replication fork. If this supercoiling were left unchecked, the DNA would overwind, snap, and the entire cellular factory would suffer a catastrophic, fatal meltdown.
Now, what's really interesting about this slide is how biology elegantly, almost miraculously, solves this massive mechanical problem. Enter a highly specialized enzyme known as topoisomerase. You can kind of think of this as the factory's elite safety engineer. Its entire existence is dedicated to racing ahead of the helicase and managing that dangerous physical tension. Topoisomerase carefully controls and relieves the extreme stress caused by the unzipping process. It does this by essentially snipping the DNA, allowing it to uncoil just enough to relieve the pressure, and then seamlessly gluing it all back together before any permanent damage is done. It's a spectacular, highly precise solution that ensures the entire replication assembly line doesn't literally tear itself apart.
Let's move to and see how this builds, because this molecular factory doesn't just run non-stop, totally out of control. It has to be turned on at exactly the right time. And there's a fascinating plot twist to how this all starts, based on recent research. Before replication even begins, the machinery sits quietly in a pre-replication state, resting as an inactive unit known as the MCM27 complex. It's essentially dormant, just waiting for orders. But when the precise biological signal is given, a breathtaking transformation occurs. This inactive complex is sparked into life, transforming into the active CMG helicase. This is the ultimate spark of initiation. It is the exact moment the factory flips the on switch and begins the whole magnificent, chaotic process of DNA replication.
So the crucial point is when we zoom out and look at the big picture of how your DNA is copied, it's clearly not just a random list of biological events bumping into one another in the dark. It is an awe-inspiring, rhythmic masterpiece. We have the MCM 2-7 complex flawlessly initiating the process from its dormant state. We have Helicase burning through ATP to forcefully unzip the double helix. We have those single strands standing by perfectly to act as exact templates. And we have topoisomerase running ahead, expertly controlling that potentially lethal physical tension. It is a perfect, synchronized dance of controlled molecular processes and precise enzyme coordination.
Which brings us to a final, rather humbling thought to end our explainer today. We've just seen the sheer perfection, the coordination, and the blinding speed of this microscopic factory at work. But consider this. What happens when this otherwise perfect biological machinery makes a single mistake? Just one misplaced molecule among billions. The fragility of this system is honestly just as fascinating as its perfection. That precise molecular coordination is happening right now, inside you, constantly working flawlessly just to keep you alive and functioning. But it leaves you wondering, how does the factory fix a typo on the assembly line before it causes total chaos? Think on that. Thank you so much for exploring the molecular mechanics of DNA replication with me today. Keep wondering, keep examining the world around you, and I will see you in our next explainer.